Everyone. I think we have a very nice group joining the session this morning. My name is Kerry Mulhadwa and I am from the Northwest University. And today I have the honor of presenting to you my first year master's student, Ms. Hesmarie Botha. She is currently doing a joint master's between NW and SAIAB. She's enrolled in environmental sciences. She has completed her undergraduate degree in biological sciences and zoology and microbiology, and has also done her honors degree at NWU in environmental sciences with aquatic health, ecosystem health. So today, Hesmarie will be speaking about her project on a really interesting group of parasites. They are marine parasites called gnathiids. So they are often referred to as mosquitoes of the sea due to their similarity in feeding habits, but I will let Hesmarie elaborate on this in her talk. And Hesmarie is still in the beginning stages of her project, as you will see, but she's very excited to share this project with you. So before we begin, I would just like to remind everyone that if you have questions during the talk, you can type them in the chat and we will deal with them afterwards. Otherwise, you can ask your question directly at the end of your presentation. So with that, I will then hand over to Hesmarie, who will hopefully share her screen.
Thank you, Prof. I appreciate it. Goodness. Sorry, something happened to my screen and I'm not sure. Just give me a minute. There we go. Can everybody see my screen?
Yes.
Thank you so much. Okay, so good morning. Thank you for the opportunity that you guys have given me. Sorry? Yeah.
We're seeing your presentation notes.
Oh, goodness. Okay. Sorry. I don't know what happened. Something went wrong in the meantime. Just give me a moment to fix this. Let me try this. Is it sharing the right one now or not?
Perfect.
Great stuff. Okay, sorry. Me and technology don't see eye to eye sometimes. But I am very excited to share a bit on gnathiids with you guys today. I have developed quite a passion for them throughout my honors year, and yeah, the more I learn about them, the more interesting they get. So I hope I can teach you guys a bit about them today. As Prof. Kerry mentioned, I'm still in the early phases of my master's degree, so I don't have as much results to show yet, but what I have, I will share a bit on. But I'd like to give you guys some background to what gnathiids are and yeah, a bit on their life cycle and so on. So moving on. Oh, goodness. Oh, there we go. So what are parasites? Parasites are organisms that live in or on other host organisms and obtain their nutrients from the host at the host's expense. This group is considered to have a highly successful lifestyle as they parasitize all taxa that follow free-living lifestyles. You also get, or there are also cases of hyperparasitism, which occur when parasites target other parasites. Parasites attached to hosts found in terrestrial, freshwater, and marine environments. However, terrestrial and freshwater organisms are better documented, largely due to the challenges associated with marine sampling. So these challenges include the depth of the ocean, restriction to the use of research vessels and nearshore or intertidal zones, and the need for advanced equipment. It's more expensive, so you need funding, and you need an experienced personnel if you want to attempt to sample from deeper waters. So among these marine parasites are isopods known as gnathiids. They are found in shallow intertidal zones along the coastal marine environment, as well as in depths of up to 300 meters or even deeper. Gnathiids are considered protilium parasites, which means that the juveniles are the ones that feed on host organisms for nutrients, while the adult stages are free-living and benthic dwellers, which mainly focus on reproduction. So elasmobranchs, which are sharks and rays, as well as teleosts, which are your ray-finned fishes from various families, have been identified as suitable hosts for these parasites. Some hosts include your gobies, your rocksuckers, your black-spotted torpedoes, puff-adder shy sharks, your horned rock skippers, convict surgeons, black-spotted surgeons, cloud wrasse, and your leopard cat sharks. Just to list a few, these also along our coast, so there are other hosts from various other continents as well. Still on the other note is that gnathiids are also, they parasitize a wide diversity of species. So there is no, let me rather put it like this. They have been shown to parasitize a wide range of species, but they also have some species specialization, which means that they have shown some preferences for hosts within specific families. And although we do know a bit on the host, we don't know everything about hosts from all the species we have currently documented. There have been some that have been described from a free living environment, so we have no idea what their hosts are, while some of the others have a very long list of hosts that we have found them on. So just to give you some context there. And then basically gnathiids infection does not always lead to host death, as the likeliness correlates between the size of the host and the severity of infection. And what I mean with this is gnathiids don't necessarily, if they have, or if a few occur on a host, they don't necessarily kill the host if it's a larger fish. But with fry, newly hatched fry, there have been cases where as little as one gnathiid infection could lead to the host death. So yes, size in the situation, the load of infection and so on, do have a role in that regard. So they also have some other negative effects. Studies have found that they can reduce the hematocrit level, which basically suggests that there's a reduction in red blood cells. They've also been found to increase the stress hormones such as cortisol. The infection has led to compromise of the immune system, which isn't exactly ideal in any situation. They're also associated as vectors for blood parasites between hosts. And this is where they get a bit of similarity to mosquitoes, and why they have also been linked or called mosquitoes of the sea. And these vectors, or similarly to mosquitoes that transfer malaria parasites, it's basically a similar situation. So although most of the negative effects are more prominent on teleosts, likely related to their size, they have been found to negatively affect elasmobranchs as well. It was found that infection in the buccal chamber of some stingrays disrupt the epithelia, which refers to your cover or your surface tissue, as well as the smooth muscle of the host. And they also caused inflammation of the dermal or the skin tissue. So furthermore, excessive mucus production and blood congestion were also noted at affected areas brought on by the physical damage caused by gnathiids. They also affect gill filaments through lamellar disruption in some sharks. And in the image there, you can see where they're attached to the gills on the lamella. Although the presence of gnathiids does not appear to have a drastic effect on the elasmobranchs, even when they are abundant, the damage they cause appear to suggest a prolonged association between the host and the parasite. So these temporary ectoparasites start their first stages of their life cycle as a segmented zoophia that use their needle-like mouth parts to touch the gills, nares, which are the nose, irons, lips, buccal cavity, the cloaca, fins, and external body surfaces of their host. In the cases of elasmobranch hosts, they have been found to attach to claspers as well. They consume blood and other fluids such as tissue fluid, lymph, blood plasm from the host, and both their feeding methods and their nourishment preference has been used to compare them to feeders. So the gnathiids feed until the thorax becomes swollen, so that middle section with the blood meal, after which they are classified as Breneza in the first stage. And you'll see generally, just for short, we label them as Z1 through to Z3 and P1 through to P3. So as they are crustaceans with an exoskeleton, they need to molt to grow to the next stage. Therefore, once sufficiently fed, they will detach from their host and hide in sponges, coral, mud, and other substrate, where they digest their meal until they are ready to molt into the next segmented zoophia stage, as you can see over there. So the process repeats 3 times until the gnathiid juvenile reaches the 3rd Bernese state, which will, where it will molt to either a male or a female then. And it's very interesting to see in the P3s. So they'll also already start to show some of the egg development when it will molt into a female, or the neck region just underneath the head will be slightly more prominent if it's going to shift to a male. So yeah, it's very interesting. It's not always a full truth detective method for new parasitologists, because I was definitely struggling in the start, but yes, it's very interesting to see. And then feeding and molting periods differ between the species, and the time to complete each increases with each passing stage. When considering their physical features or morphology, gnathiids are unique compared to other isopods, as they do not display the usual 7 pairs of legs. Instead, they display 5 pairs of uniform legs. So you can't see all the legs because they are tucked under, but they're very interesting because they look real, like they have a lot of similarity, or they are really uniform. The only difference is that the first 2 are directed different in a different direction compared to the last 3. So then moving on, they also have, or unlike other isopods, that are morphologically similar through their life cycles, gnathiids display unique characteristics between different stages, as you have seen. And this is emphasized by characters such as the protruding mandibles in the males, and the lack thereof in the females, as well as characteristics such as the larger eyes, compound eyes we see in the juveniles. So the diverse physical features, also referred to as morphology, seen between the life stages, contributed to much confusion in past descriptions. Although some features have been linked between the adult and juvenile stages, it has still proven challenging to link them solely based on these features. Of the 3, males, females, and juveniles, the males prove to have the most distinct morphologically differences between the various species, and is why they were used more commonly in species descriptions. Although this has made the species descriptions possible, it has proven challenging to link females and juveniles to males if we cannot confirm that they are from the same species. However, taxonomic terminology and interpretation are still being adapted today. And the standards for these descriptions were established as early as Menard's publication in 1926 and continue to undergo refinement, which has necessitated the redescription of species described prior or from earlier descriptions. So this is just a bit of a representation of some of the diversity we see between different genera. So these are 3 different genera found along our coast, the one Afrognathia, the other Cecognathia, and the third Gnathia africana. And as you can see, they have some unique structures. So just an idea of some of the features we look at. As you can see, they all have their unique head shape, or the cephalosome, which also carries different features that we look at and consider with their description. Then moving on, even factors such as the compound eyes ratio in comparison to the rest of the cephalosome shapes are features that we consider with description. Then there are the mandibles that have unique shapes, features. Each of them has a different teeth size, pattern, distribution. There are other aspects such as the presence of the carina and so on that we take a look at. So these are very, basic image that I'm showing here, but you will see later a better representation of the things we look at. And then stuff like the tailton shape and the uropods and so on. So these are just a few of the many different features we take a look at. Obviously, we look at the rest of the body as well, but this was just a quick representation of everything we consider. So, according to Spalding's paper in 2007, there are 12 designated marine biogeographic realms. These realms are considered to be large regions of coastal, benthic, and pelagic ocean that share similar plant and animal life due to unique evolutionary history. They have high levels of endemism, which means that a lot of the organisms found in these regions occur naturally there and are restricted to them. The factors that influence this unique development include water temperature, historical and broad-scale isolation, and proximity of the ocean floor. The gnathiids are present in most of these designated realms. In realms like the central Indo-Pacific, which you can see there by the star, are high with tropic waters, a lot of coral reefs, and higher biodiversity. Up to 46 species from one genus, Gnathiia, has been identified there. However, on our coast, the Temperate Southern African Marine Biogeographic Realm, or further referred to as the TSA, which also boasts a unique biodiversity, we've only found 7 species from this genus, which you note is a lot less than we have found from the central Indo-Pacific. In total, we have 9 accepted species from our coast. Of these species found along our coast, 3 genera were observed, of which Gnathiia had the most, obviously. This indicated some biodiversity found within our region as well, and indicates the potential to find more. So literature indicates that 5 of these species were found at depths between 26 and 347 meters, which circles back to the challenges of sampling in deeper marine environments. Furthermore, of the 9 species, 4 were described between 1914 and 1920, which was more than 100 years ago. To date, Gnathiia cryptopaius and Gnathiia africana are the only 2 that have undergone redescription. So next in line for the redescription is Gnathiia spongicola, which was first described by Barnard in 1920. As seen in the illustration, there were limitations of the features included in the description. Although this may have been suitable for the time and the technologies available, we have since made great progress in scientific community, which allow us to observe features previously unseen. Furthermore, Barnard also described a group, Gnathiia spongicola variation minor, which he believed belonged to the same species as Gnathiia spongicola, but displayed minor differences. With the advancement in today's technology, we will be able to clarify whether this group is indeed the same species, or if we should rather consider them as a completely different species. So my project has 2 parts. For the first, my hypothesis was what is currently considered a single species, Gnathiia spongicola, is rather a species complex. And our aims for this project part is to resolve the Gnathiia spongicola species complex, and the objectives were to redescribe Gnathiia spongicola from the type material we have, as well as morphologically identify and describe other species within the Gnathiia spongicola complex. So currently, we have some gnathiids on loan from the Zico South Africa Museum, which are being redescribed and clarified, whether Gnathiia spongicola and Gnathiia spongicola variation minor are indeed the same. So this is being achieved through the use of a light microscope that is used to study the features of gnathiids. The first step is to document all features of the gnathiid through photos or photomicrographs captured by the Nikon camera, which you can see attached there to the microscope, as well as through the use of the NIS element program on our computer. These photomicrographs are of whole gnathiids as well as stained and dissected gnathiids. So the whole gnathiid you can see there at the bottom, and that's not the stained of the gnathiids I'm working on, but a previous image of one of the stained photos we have. As you can see, it brings out a lot of the features very nicely. So the staining is done through the use of lignin pink, which enhances the visibilities, as I mentioned. Which enhances the visibility of the features under the microscope. We also make use of scanning electron microscopy, or SEM, which is conducted to observe other features which aren't necessarily visible under a light microscope. So these features include the presence of scales, such as the pectinate scales observed on the pleotelson in the image, which I've put there for you guys, or the presence of hair-like structures called setae. So there are other features we look at as well, but that's just an idea of things we can't necessarily always see under the light microscope. And it just gives you a more thorough description at the end of the day. Gnathiid is then drawn using the camera lucite, which is attached on the microscope there, which you can see. And finally, an ink drawing is made using Adobe Illustrator and a drawing tablet. So as you can see from the example plate of Gnathiid apipiendae, which is actually done by both my supervisors. Sorry, I forgot to say, my supervisors are Prof. Kerry, which introduced me, and Prof. Niku. So Kerry Malherbe and Prof. Niku Smith. And sorry about that. And it is evident from this plate that they produced in their paper, and that there are a lot more features that we take a look at. So yeah, I don't know if you could tell based on the image I put on earlier, but there are a lot more details that we consider now and that we can see and incorporate into a description, which makes it, I think, more thorough. So by comparing the two, the plate on the left representing the original type drawing and the photographs on the right of the syntypes observed and the images that I took, I'm still busy with the description, and I can only point out the minor details from the original Gnathiid spongicola description. So features like the mandibles and the legs and periopods were completely omitted from previous descriptions. As seen from the micro photographs, the mandible, its structure and the number of teeth are far more visible on the imaging we have now compared to what was represented on the original paper. As well as when you look at the periopod underneath the mandible, you can see those spines on top of the leg, which are very prominent and really cool under the microscope as well. So these were also not included in the paper. So these are just some of the features that allow us to make a very more thorough taxonomic description and what we hope to achieve in our redescription there. After we have collected all of the information regarding the taxonomical features and from the drawings we've made, all the information is placed in the descriptive language for taxonomy or Delta program and used to generate a standardized description. The first aim for the section of the project is to redescribe Gnathiid spongicola, as I've mentioned, and thereafter we can clarify the placement of Gnathiid spongicola variation minor, which is currently underway. So most of the drawings and description has been completed from Gnathiid spongicola's type material, and then I will follow up with clarification with variation minor. So the second part of the project aims to produce a link between these species, along with the ambiguity of Gnathiid morphology, as well as the solo growing list of accepted species, or even fewer reports on relationship between these Gnathiid species, both from a morphological point of view and a molecular point of view. So this is achieved through the use of phylogeny, which aims to understand and produce the evolutionary history and relationship between different organisms. So a comprehensive understanding of their morphological and molecular similarity or differences may provide links to better comprehending these isopods. However, the last extensive morphological phylogenetic analysis was conducted by Cohen and Poor in 1994 and was only incorporating gnathiids from Australia. To date, no publications have incorporated a molecular phylogenetic tree, as there has been a lack of published molecular data of gnathiids. This has produced a need for more thorough phylogenetic analysis of gnathiids from around the world and specifically from the TSA. So this puts us in the second part of my project, and the second part is the morphological phylogeny and molecular phylogeny of gnathiid species from the Temperate Southern Africa marine biogeographic realm. Will follow similar phylogenetic patterns is what we believe to be the hypothesis. And our aims for this is to conduct a morphological and molecular phylogenetic analysis of all gnathiids from the TSA. Our subsequent objectives are then to collect gnathiid isopods from the TSA to sequence all of these fresh gnathiids and then to produce morphological and molecular phylogenetic trees, especially from the TSA. So to achieve this, we will need some fresh material, and this is just a representation of some of the previous collection methods. So some of the methods that are used target host species, and this is done using a rod and reels or hand nets or baited traps. And these methods are generally restricted to the intertidal zone. You can do these methods in deeper waters, however, okay, not the hand net, but with the rod and reel and so on. The only restriction are you need more funding, a boat, personnel, and so on. So it's, yes, there are restrictions, as I mentioned earlier. So how this would have, or that usually works, let me put it that way, is host organisms are captured from their marine environment using one of the previous methods mentioned. Thereafter, one of two parasite collection methods is followed. So in the one hand, host dissection occurs, and then we generally do a thorough scan for all parasite groups found on the host. They are then isolated and preserved in their various methods. And then on the other is we isolate fish or the host species in their own tanks with fresh seawater, and they are left for a period, generally overnight, to drop gnathiids in the water. So the gnathiid will attach, feed, and then detach, as I mentioned earlier. The water from the host environment is then scanned for the gnathiid, and any gnathiids found are then isolated and kept in seawater. They're generally sorted and preserved as specified. But what happens is generally we isolate the P3s and we'll try to get them to molt into the adult stages. So this generally takes a bit longer. So for the following days that the gnathiids have been kept in an isolated jar, they will receive water changes and monitored to observe when the gnathiids molt into either the adult males or adult females. And then we preserve them in 70% molecular, we preserve them in 70% ethanol if we want to use them for morphological characterization, and in 95% or ethanol or higher if we would like to do molecular sequencing on them. So although these methods have proven successful and still prove successful, they are still being used. Another method for collection was developed by Mary Shadipa and a research team and was published in 2023, which eliminates the need to obtain the host. So this method makes use of light traps. And as you can see there, it uses relatively inexpensive material. So PVC pipes are sealed off on one end and have a screw cap lid on the other end. On each side of the pipes, there's a clear funnel that's placed in, which allows for an entrance into the trap. And the geometry of the funnel makes it harder for the gnathiids to exit. So there's not a very high likeliness of that. Then as gnathiids are or lured by light, these traps are placed at night, and we use a diving light to attract them. So they are placed in shallow nearshore environments, preferably at a lower tide. And then we'll place them prior to the evening and then collect them once they have been out for a sufficient period. So they are kept in place with the weights that you can see there. And then we also have markers to find them again. With our dynamic coast, it has proved a bit interesting to get the traps to stay in place, but we are working on refining the method. Upon retrieving, funnels are sealed with clay to ensure that all organisms remain trapped, after which the trap is decanted into a bucket when we are at a more suitable location. And then all the organisms are scanned through, and any gnathiids found are then isolated. And depending on what we want to do with them, we will either then preserve them for morphology or molecular, and in this case, preferably molecular. So for the phylogeny bit, both morphological and molecular phylogeny analysis will be conducted. For the morphological part, specific features will be considered and compared between various species. Those with most morphologically similar are more likely to be closer related and will be closer on the phylogenetic tree compared to those that share fewer features. And then with the molecular phylogeny, we will be collecting DNA from the freshly collected gnathiids, which will then be extracted. We select genes which we then try to amplify and sequence. And these sequences are then compared to or between species to establish which are more closely related or don't share as many features and traits. So what has been done and what is next? So Gnathia spongicola description is almost complete. And once that is done, I will be moving on to Gnathia spongicola variation minor. There is also an isopod sampling trip planned soon. So hopefully we will get some fresh material that we should be able to sequence from that. And then once all the sequencing is done, which I expect should be done by the end of the first quarter of next year, I can use all that information to move on to constructing the phylogenetic trees rather. So there's still much to learn about gnathiid isopods. The deficiency of previous standards in taxonomy description has necessitated redescription of species such as Gnathia spongicola and caused some confusion with groups such as Gnathia spongicola variation minor, which needs some clarification. Furthermore, there is much room for growth regarding morphological and molecular links in species, especially from the TSA gnathiids. So I hope to address these gaps in the next part of my master's. And I believe that the information we gather will help contribute to what we currently know of gnathiids and hopefully create a foundation for future studies as well. So thank you so much for your time. And if anybody has questions, please feel free to ask.
Thank you very much, Hesmarie. That was a lovely presentation. I think we'll start off with the chat questions, if that's fine. So Vanessa asked, are there environmental cues that decide if the gnathiids molt to become male or female?
So I am not sure specifically about environmental cues. What has been noted for the molting is some species have a specific number of days. So I think with Gnathia africana, it's like 17 days before females will molt from a P3, 16 or 17 days before they molt from a P3 stage to the adult. So I'm not 100% sure on the males number. But yeah, so for the environmental specific, I do think there's potential for it, but from my knowledge, it's more a factor about the time, I think. And then amount of time they get to digest. Prof. Gary might be able to add on to that.
So there is another question that's related, which I'm just going to go to quickly. That's Francesca's. It says, have there been any records or observations of changes in range among bordering realms due to changes in the environment?
So I have not specifically focused on any environmental changes study, as my focus is largely on the taxonomy. So in terms of ecology and so on, I still have a bit of limited information. At this point in time, I believe as I study further, I will be exposed to more knowledge. So I cannot answer that question at this stage.
Okay. And another comment from Vanessa, or another question, was the male seems to have more pronounced mouth feeding parts. Is this correct?
So it's the mandible, yes, is 100% more pronounced on the male. As I don't really feed, I don't think the feeding part specifically is more pronounced. But yes, no, the mouth parts are one of the features I think that make them more distinct and ideal for taxonomic description. So yes, I would say they are definitely more pronounced.
Fantastic. So there are some other compliments in the chat for you that you can look at, in a nice paper that you can look at for sturdy rocky shores light traps, which will
Thank
be
you.
good to know.
I'll definitely take a look at that.
Are there any questions from the floor? Anyone? I don't see any hands raised.
Can't see anything on my end.
Oh, there's one. Francisca, yes.
No, I just wanted to further, well, first of all, well done, Hesmarie, really clear presentation. In relation to the extension range, it's not about the ecology, it's just that maybe wherever you're sampling, you might just start getting perhaps more species or different species from what you've listed in relation to that biogeographic map that you present, and simply because especially at the edge of a biogeographic region, you might just start having some overlapping with other regions, and hence the presence of potentially different species.
Sorry, was it a question or a statement?
It was just to further comment as that my
Okay.
question may have seemed an ecological one, but I think it's quite linked to your taxonomic investigation.
That does make sense. I do believe there will be some overlapping, and I think in places like Australia, they have definitely seen a lot more overlapping within smaller units as well. But yeah, I think we definitely have a lot more room for growth along our coast. Some of the areas have been sampled a lot more, I think, than ours as well. So yes, hopefully the future will unveil a lot of interesting new discoveries.
Nika.
Thank you, Kerry and Hesmarie. Great work promoting the gnathiids.
Thanks, Rob.
It's a very interesting question about the ecological range in the different provinces and regions. So I can maybe just add what we have found is that the dominant intertidal species from the temperate region is Gnathiid africana. But then actually Kerry did describe as part of her studies, a species from the east coast of South Africa, which is in a different area, and Gnathiid pilosus. And then when we started to work on the Eastern Cape Coast, Sinsa, and even they're close down to Grahamstown, Bookness, and those kind of areas, we actually did start to find the overlap between Gnathiid africana and Gnathiid pilosus. So also just showing that these borders do shift depending on what's going on. And I definitely think if with the change in temperatures and those kind of things, we will see those borders shift. And so theoretically it must be there, Transkei somewhere. So we were collecting on the temperate part of the coast still, and we started to find pilosus. So yeah, so it's quite interesting when you look at these intertidal gnathiids as well, and they do tell a story. Thank you.
Thanks, Rob.
Any other questions? Hesmarie, maybe you can elaborate a bit more on how many species do you think you can expect to find within the species complex?
So I believe we have the potential for at least 2, just based on the fact that Barnard actually noted quite a few variations in his own description of Gnathiia spongicola variation minor. So I think it's, I don't want to say safe to say, but optimistic to think that we'll at least have one additional species from the species complex, I want to say. So yes, but we have some potential other species lined up. It's just unclear at this point.
Any other questions?
There's a question in the Q&A.
So there is a comment from Francesca saying there is scope for a PhD looking at thermal tolerance of these species should they overlap or extend range.
That could be really interesting.
Just adding more. Lots to do.
Gotta first survive this one. But yeah, no, that could be very interesting, especially because when I spoke to Prof. Paul Sickle, I do believe temperature does have some influence on the different species we find, especially for along our coast, the light traps in the warmer tropical regions where they put the light or have successfully put some light traps. They cannot leave the light traps for prolonged periods because the gnathiid metabolism works a lot faster there in the warmer waters and the warmer regions. So they actually need to remove the trap within a few hours just to ensure that the species stay alive. Where on our coast, because the water's a bit colder, metabolism is a bit slower, so we can actually leave the traps a bit longer. So yeah, I think it would be interesting to see what the influence of temperature is on these interesting little creatures.
Okay, we have another question from Matsubani.
Thank you. So I put a question in the chat. I'm just going to read it quickly in the Q&A. Sorry, not the chat. So I wanted to ask if there is any mitigation strategy for the pests, because I heard how you mentioned some fry do die as hosts. So I was just wondering if there's any kind of research on potential mitigation strategies, especially when thinking about infections on endangered elasmobranchs, because we are also expanding MPAs to protect elasmobranchs, and if we are putting them in this sanctuary and they passing around this pest, that could be futile, you know. So I'm just wondering if there's any kind of mitigation strategy out there.
So that's a very interesting question. And yeah, it depends on, well, no, it doesn't really depend on, but as I mentioned, they don't always lead to death. So it's more a case of that they have some negative effects rather than always lead to death. I think they are not very documented on aquaculturally important fish, if I'm not mistaken. So I don't think there was immediate research or any large-scale research done on trying to manage gnathiid infection. And as sharks are slightly, well, a lot larger than fish fry, I don't think that a newly hatched shark, if I can put it like that, or a young shark would succumb so easily to gnathiid infection, just because they are a lot larger in size, and just when comparing the ratio to new fish, they have a lot more blood and other fluids for the gnathiids to feed on before they are affected. So I don't think they would immediately be able to contribute to that, although it would be interesting to expand on that, I think. I don't know if you have something else to comment on that, Prof.
Oh, sorry.
Yeah,
No.
okay.
Okay.
All right. So I was just asking if there's any, like while you were doing your readings, your literature search, if there's anyone looking at how these pests could be, I don't know, because I've watched Bragg videos and I've seen some wild fish with infections and others with dying tissue and frilled fins. And, you know, so I'm just thinking out there in the wild, if there are these infections, is there anyone looking at a way to curb these infections or pests? I don't know.
I think the research is at the moment just trying to expand on our knowledge of the gnathiids at the moment. So I'm not sure specifically, but I see Prof. Nico has his hand up, so he might have some comments on research to try and control them. So Prof.
Hi, yes, thank you for that comment. I think just one thing that is always interesting when you do look at the parasites is that in the natural environment they're not normally pests. So usually only when the environment is under stress, then you have a problem with that. So actually, in fact, we're busy with a study looking at the parasites within and outside of marine protected areas, and the parasite populations are very healthy and quite diverse inside a marine protected area, showing that marine protected areas actually do protect this, what we call the hidden biodiversity as well. So in any normal healthy system, you will find a normal distribution of parasites, high diversity, and then you'll have just some hosts with high infections in order to sustain the population. But in general, they don't, in natural wild healthy ecosystems, they're not really considered as a pest. In terms of gnathiids specifically, the only place where there has been reports of them causing damage and death to hosts is in aquariums. And then there's quite a number of reports there where the moment they get into the aquarium in a confined space, then they can cause a lot of damage. And for that, they do the normal control that they do for crustaceans using copper and things like that. But the most important thing is to break the life cycle. So that is what the work that Hesmarie and others in our group do on the life cycle of these organisms is very important. So once you sort out our life cycle, you need to break that. And then that is the way of the best way of protection. So yes, I hope I could add some ideas to your question there. Thanks.
Yeah, it does. Thank you so much.
Thanks, Prof.
And if I can also just add to that, everything, they are small and they're only effective, they only cause a lot of damage when there's large numbers in agriculture. So most of the time they are completely harmless, and we can sometimes even have multiple species on the same host. So we had elasmobranchs in Australia that had 2 species on one host, and the host was completely fine up until we had to kill it. Other than that, they're not affected. Any other questions? You did a good job, Hes. There's
Well, I confused everybody.
They're all stunned by this cool little isopod.
Maybe.
Oh, well, in that case, if I will give it going once, going twice, if anyone's got any questions.
There's a
There's
hand.
another one. Prince. Sorry, Prince, I don't know if you.
Oh yeah, it's not actually a question. It's just I wanted to ask if I can get this recording actually.
I think it should be Lucky should have that information, I think.
Yeah, if they can advise whether I can get this session as well, just so that I can sit down and listen to it again thoroughly as well. So because before I draft things that are not accurate, so just
Yeah,
so that
fair
I can get all the facts together. So if possible, I can drop them emails because they've shared their emails, so I can do that, and then maybe they can provide me with the link to the session.
I believe Lucky has the information regarding where everything's posted.
Yes, correct. The recording will be available on the SAIAB YouTube channel. I'll share the link on the chat.
Right, there's one
Okay,
more.
thank you.
Thank you.
Sorry. There's one more comment in the chat, and that was from Anya saying we also have colleagues doing more ecologically focused studies in the Caribbean. So that's Prof. Paul Sickle. His group is doing ecology everywhere, the Caribbean, the Philippines, and doing fantastic work. It's also available online for all the papers, so you're welcome to get a look. And they have a little group called Gnathiid Nation, so they're very, very cool. But yes, if there are no other questions, then I would just like to thank Hesmarie for, again, another lovely presentation. And thanks, SAIAB, for this opportunity for her to share her work. And I would like to wish Hesmarie all the best for the rest of her masters. And thank you, everyone, for your attention and your questions. And I hope you have a good day. Take care.
Thank you.
Goodbye.
Bye.